Hello, everyone. My name is Rose Wayne. I am a PhD student in the Maruska lab at Louisiana State University. Thank you for coming to my talk, The Neural Mechanisms of Social Defeat. Studies in many species, most commonly rodents, have demonstrated that social defeat is a powerful experience that can lead to drastic changes in physiology and behavior, many of which are negative, resulting in important consequences for reproduction and survival. Repeated social defeat results in reduced reproductive fitness, sickness, and behavioral abnormalities. The neural circuitry involved in fear and anxiety is highly conserved throughout evolution. Animal models can be used to better understand the impact social defeat has on how organisms cope with social defeat stress. This includes humans, with one of the most prevalent forms of social defeat being bullying. Animal models could also be used to improve human models of psychiatric disorders such as anxiety and depression. This leads us to the model organism I will be sharing with you today, Tilapia burtoni. The cichlid fish, Tilapia burtoni, are highly social fish native to Lake Tanganyika in eastern Africa, and their plastic behavioral repertoire is well characterized. Males exist in a dominance hierarchy along a continuum, but are characterized as either dominant or subordinate. Social interactions that include both defeat and rises in rank are an integral part of their lifestyle, making them an ideal model to examine the neural correlates of social defeat. On the top, dominant males are brightly colored, defend a territory, court females, and are reproductively active. Depending on their social environment, they can switch their social state many times throughout their life. On the bottom, subordinate males are drab-colored, shoal with females, do not maintain a territory, and are reproductively suppressed. Here's a quick video showing aggressive territory displays, lateral displays, where they sort of waggle their bodies and push water at one another, bite, and flare their opercula, also known as their gill coverings. Dominance is the default state. If a dominant male is absent or removed from a population, a subordinate male will rapidly transition to the dominant phenotype within minutes. If a dominant male is subjected to social defeat, often by a larger, more dominant fish, they will rapidly lose their coloration and transition into the subordinate phenotype. This leads to my research aims. The first, identify if repeated social defeat results in long-lasting behavioral changes of coping strategies. And two, identify the neural and behavioral correlates of repeated social defeat over time. I've developed an extended repeated social defeat experiment using a resident-intruder paradigm to record and study the defeated fish's coping behaviors over time. Experimental fish are acclimated in their territories. We have intruder and a resident territory. And then these intruder fish are chronically socially defeated for 4 days. Each day, the intruder male is placed into the resident male's territory, who is larger, for 30 minutes, where he is defeated. Afterwards, he is placed back into his territory, given a 4-day break, and then after they are re-exposed to a normal social defeat condition with a new resident aggressor male. Control fish are also acclimated, but then not chronically socially defeated for 4 days, so they're placed into the resident's territory by themselves. Afterwards, they are given a 4-day break, and then they are exposed to a normal social defeat condition with an aggressor resident male. With my experimental design, I observed and recorded the defeated fish's coping behaviors over time. Here's a quick rundown of the types of behaviors recorded. Proactive fish are more bold. They respond with aggression or attempt to escape the stressor. Conversely, reactive animals are described as shy and do not attempt to escape the stressor. Data shows that repeated social defeat leads to a retention of reactive coping behaviors. On day 1 through 4, control fish display no coping behaviors as they are not responding to an aggressive resident. But control fish on day 9, after the 4-day break, experience an ethologically relevant level of social defeat, and they spend a similar amount of time performing searching and freezing behaviors. Just a quick reminder that searching is a proactive coping behavior and freezing is a reactive coping behavior. Chronically defeated fish can easily be divided into 2 different behavioral groups, susceptible and resilient. Chronically defeated fish on day 1 in both groups display a similar amount of time performing searching and freezing behaviors, similar to the control fish on day 9. During the course of repeated social defeat, especially on day 3 and 4, susceptible fish freeze for a significantly longer period of time than searching. After the break, susceptible fish retain the pattern of freezing for a significantly longer period of time than searching, indicating persistent effects of social defeat on their coping behaviors. On the other hand, resilient fish did not seem to change or retain one coping strategy over the other after time. These results are promising because they are also similar to what is seen in other ethologically relevant animal models, such as rats and hamsters, where defeated animals often avoid social interaction, increase submissive and defensive behaviors when approached by a non-aggressive or aggressive conspecific. It's important to note that maintenance of a fixed reactive coping strategy over time in A. burtoni is a behavior outside the expected social plasticity in this species, which could negatively impact their health and fitness. Since behavioral results demonstrate that repeated social defeat has lasting negative impacts on future behavior, I also wanted to see if it's associated with distinct neural activation patterns in the brain. I stained behaviorally relevant regions of the brain, such as the social decision-making network. The SDMN is a conserved neural and gene expression network that are the proximal mediators of most social behaviors. I performed immunohistochemistry for the activation marker phospho-S6 ribosomal protein, also called PS6, followed by quantification of activated cell densities in these candidate brain nuclei. I have an example of a PS6 stain for a control and treatment fish whose brains were collected after day 9 of my experiment. In the ATN, the anterior tuberal nucleus, found here in the diencephalon, which is also found to be associated with proactive coping behaviors. In our control fish, we see some activation, but not nearly as much as in the treatment fish. In the SR, the superior raphe, found here in the hindbrain, it contains a population of serotonin neurons associated with reactive coping behaviors. It may also have conserved function in mediating reactive behaviors associated with social defeat across vertebrates. In our control fish, we saw a lot of activation, but almost none in our chronically defeated fish. These stains matched behavior exhibited by these individuals. The control fish was much more reactive than proactive, and the chronically defeated fish maintained an even mix of proactive and reactive behaviors. So for future directions, as we've seen, the ethologically relevant social defeat model of A. burtoni can be used to understand how defeat changes the brain and future behavior. I have behavioral and brain data for the same individuals, providing valuable insights for linking brain function to behavioral outcomes. Work will continue to be done with PS6 staining and the SDMN, and my next step will be complement my PS6 staining by using RNA-seq to look at differential gene expression levels in behaviorally relevant regions of the brain in the SDMN between my control and treatment groups, which I have already collected tissue for. And lastly, as the brain of fishes shares many properties with mammals, studying the long-term effects of repeated social defeat will continue to provide insights towards improving other animal and human models of psychiatric disorders with important societal and biomedical implications. I would like to thank everyone in the Maruska lab, especially my undergraduate assistants, Ava Karam and Alora McInnes. I would like to thank funding, Sigma Xi, and NSF, as well as all of my committee members. Thank you everyone very much.